on to the second half of this presentation, which is with Biosortia and their microbiome mining, which was new to me when Ross first reached out to me. So I had to buy a book and read about microbiomes along with all the papers that Ross sent me. So I am not a microbiome expert, but I got a very rudimentary understanding of what they are and what they can do with them. And this is really exciting. I believe that there are a lot of opportunities here, and I'm going to turn it over to Ross. Take it from there.
Terrific. Eugene, can you hear me okay?
I can.
Excellent. Well, then I'll jump right in. I appreciate the opportunity. Thank you, Eugene. Thank you for Polyplexus and technology ideation. What we're going to talk about today is industrial scale microbiome mining and how we could potentially collaborate not only with the government and the various agencies within the government, but within industry and within science and academia. Reality of it is what you're going to be introduced to today has never been taught in any academic institution and never practiced. The problem is when you get new life science technologies emerge on the scene, there's nobody to actually engage. This is as you will see, a potentially game-changing, profound innovation going to have massive impact on the data of the dark matter of biology in the microbial world, as well as direct access to what is completely hidden molecules, cellular signaling molecules, and the products of microbes. So I also wanted to point out that we've been invited to do 2 more presentations, one on the 28th of February. It'll focus on therapeutics, health, and nutrition, and one on the 27th of March. It'll focus on sustainability, environment, and agriculture.
Cool.
So what we're talking about here is an important concept. So those first 10 or 11 slides talk about the background that's important, not only for the scientists to remember from early studies of biology and microbiology, but for everyone else to put in context. The first microbes on the planet, the first life on this planet, could have been occurring as much as 4.5 billion years ago. The only reason they can't say that positively is the fossil record's been completely destroyed. But the reality is that for the next 3 billion years, microbes terraformed this planet, took a hostile, basically chemical soup of methane and no oxygen, and turned it into the Earth we have today. So during that period of time, when you think about the microbes that populated the Earth and actually terraformed it, the amount of chemical calculating that had to occur, considering the lifespan of microbes and how quickly they mutate, was really what created all the chemistry on this planet, at least at that stage. Now, when we talk about the reality is that microbes invaded nearly all habitats at that time, but ultimately evolved into all life. And today, it's not only on life, it's in life, and to a serious degree. So microbes on this planet, other than woody biomass, actually outweigh all other biomass combined. That's an important concept because that really says we're still a microbial world. Now, the human, just to put that in perspective, the human has 40 trillion microbes in it and on it, yet we only have 30 trillion cells. So we're actually outnumbered. And when you talk about the genes, everyone's heard 23andMe or 23,000 genes the human has. Well, the human microbiome has 25 million genes. We're outnumbered, we're outcalculated, and the small molecule chemistry that's produced is extensive. In the vast majority, we have no idea what it is. And by vast majority, probably 99.5% or more of the chemistry going on from microbes in the human body, science has no idea what that is. When you talk about production of food, we're all familiar with cheese and yogurt and bread, the kind of things that directly are created with involvement of microbes. But quite frankly, everything that we eat or consume, whether they're animals, whether they're plants, all have microbiomes and all play dramatically into the health and welfare of that entire chain of the food supply. When you look at microbes themselves, what makes them so powerful is they can actually move their genes to each other and to other organisms. So in the human, when you talk about epigenomics, you talk about the changings of your own genome as you age. A lot of that is because of the microbial interactions through horizontal gene transfer. So the power of microbes to actually interact and calculate solutions to things is somewhat unbelievable. When you take just E. coli on this earth, it's in massive quantities. But if you try to look at the calculating power of E. coli just on its mutation capability and compare it to the fastest, largest supercomputer, you would have to have that supercomputer in quantities that would fill up the entire state of South Carolina to equal the computing power of just one species of microbe on the planet. Now, I'm going to show this video real quick, and it'll help you understand the power of calculation.
So what we ended up building was basically a petri, except that it's 2 feet by 4 feet. And the way we set it up is that there are 9 bands. Base of each of these bands, we put a normal petri dish thick agar with different amounts of antibiotic. On the outside, no antibiotic. Just in from that, there's barely more than the E. coli can survive. Outside of that, there's 10 times as much, 100 times, and then finally the middle band has 1,000 much antibiotic. Across the top of it, of course, thin agar that bacteria can move around in. The background is black because of ink in it, and the bacteria appear as white. First, see they spread in the area where there's no antibiotic up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic. It spreads until it starts to compete with other mutants around it. When these mutants hit the next boundary, they too have to pause and develop new mutations to make it into 10 times as much antibiotic. And you see the different mutants keep this at 100. And after about 11 days, they finally make it into 1,000 times as biotic as the wild type vibe. And so we can see by this process of accumulating successive mutation that bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short period of time.
The reason I bring that particular video up is it shows you how powerful the calculating power of E. coli is to defeat an antibiotic in 11 days in a petri dish. Now, what I find interesting, and others should find this interesting, is as extensive as microbes are on the planet, as easy as they are supposedly to grow, the reality is less than 1% of the microbes will grow in a culture. And if you can't grow a microbe in a culture, getting at the chemistry it actually produces is virtually impossible. That's part of the challenge that exists out there. Culturing was done early in the drug discovery, agrochemical, other life sciences discovery, and they ran into a wall because as easy as it is for microbes to fill all habitats, in a laboratory environment, it's actually fairly hard to grow microbes, let alone induce microbes that will not culture to grow. And if you can grow them, will they even have a reason to produce their interesting metabolites? So even though science has only had 1% access to the microbes essentially as a possibility, it's delivered more than 50% of the drugs on the shelf directly, indirectly, or inspirationally. So culturing worked, but culturing stalled. And it stalled for many reasons, including you couldn't grow everything. And when you look at the next technologies, that were going to be, let's call it, the shiny star of technology, combinatorial chemistry came about after culturing microbes stalled as a way to get at the molecules in nature. Years, decades were spent doing combinatorial chemistry, billions of dollars annually worldwide, and only one drug got approved. Now, why would it that microbes can produce molecules that impact cellular signaling, and we, with the best chemistry kits in the world, essentially couldn't find those kind of molecules? Part of it is scale. When you think about the possibilities of drug-like molecules, believe it or not, there is potentially as many drug-like type molecules that could be made as there are atoms in the universe. The number is so high, the odds of stumbling upon something is absolutely next to impossible. Nature had that 4.5 billion years to try the chemistry that worked, retain the chemistry that worked, perfect the chemistry, and build other life forms. So if you want to get at the chemistry that impacts cellular signaling, whether it is in any kind of life science opportunity, the starting point is really life and microbes on this planet. That's the message. Now, certainly today's indirect technologies, whether we look at genomics, synthetic biology, synthetic chemistry, metabolomics, all of those are trying to indirectly find that kind of chemistry using DNA. And the challenge with that is DNA itself is the code. It's not the solution. You take the code, you plug it into biology, and biology does what it needs to do with that code to spit out the chemistry. So eventually we'll be able to connect the dots between all of the genomics, all of the molecules, how it actually makes those molecules, and what those molecules do. But in the meantime, not having direct access to the dark matter in biology, in microbiology, has driven up costs of new molecular entity discoveries dramatically. 84% faster than inflation, essentially since the 70s. And yet the productivity of new molecules is actually going down. So it's not a very good, let's say, yield on investment. When you talk about how in the world could you get microbes and where would you get them at scale, the first thing we may look at is, well, I'd love to understand what's going on in the human microbiome. Well, the problem is really the best achievement of recovering microbiome that's active, living, and in a habitat has been in gram quantities other than feces. Now, feces comes with the entire waste stream. There are microbes in there, but it's the decomposition of microbes and the metabolites that it's occurring. It's not the middle of the process or the beginning of the process or an active, living, breathing microbiome. It's waste products. And the same thing has been able to be looked at throughout water and wastewater technology or sludge or biofilms. The reality of it is if you don't collect those microbes in a living, breathing, active environment where the microbiome is thriving and the species are thriving, rather than in the degradation environment of waste streams, you're not really looking at the molecules that are needed to be looked at. And that's part of the problem. And when you look at filtering and processing microbes, they're so small. The viruses actually outnumber microbes 10 to 1 traditionally. So you fill up the pores of any kind of filtering apparatus. If you use chemical processing to obtain microbes, you're actually degrading it. And then the question is, how long is your dwell time where you destroy the microbes and you destroy the compounds you're actually looking for? So when we look out in the real world, this is Chesapeake Bay, and I pulled this picture off the Chesapeake Bay Foundation, so I think it's appropriate to give them a plug. The reality you're looking at is microbiomes at scale. Now, what's interesting about the microbiome you're looking at is the Terra Ocean Project proved that the aquatic microbiome overlaps the human gut microbiome 73% with genes and metabolic pathways. Now, if you can't get at enough quantity, at enough quality out of a human, and you have a 73% overlap in the aquatic environment where there is massive quantities of microbiome, you're in a position to uncover those metabolic pathways that match humans and uncover the actual molecules that could be present in humans. So think of it as a surrogate. Ultimately, we've got this massive microbiome at scale, and it could be a surrogate to many of the problems we have in life sciences in uncovering those molecules. And that's really what Biosortia does. We are a deep tech platform for access to the hidden molecules and data of microbiomes for IP portfolios. We call that industrial scale microbiome mining. The original technology was conceived internally at Biosortia, got funded by ARPA-E, DARPA, the US Air Force for biofuel opportunities. And then we pivoted to small molecule discovery and the data discovery itself. And again, we work with a lot of collaborators, but when you're dealing in a brand new technology that has never been trained, never been practiced, how in the world can a grant even be asked for? How can it be reviewed? That's one of the biggest challenges that occur in life sciences, is we've got all the specialists and experts, and none of them have been exposed to this technology. And now you have a new technology that could bring tens of thousands, potentially millions of novel small molecules into research, along with the data to support genomics and other research, yet it's unheard of and unpracticed. That's the biggest challenge with the new technology. So what is it that Biosortia really will do? We don't intend to do what others do. We intend to support them. So the main goal of Biosortia is to do the mining of the microbiomes, the collecting of the data, the building of the data sets, the advancing of the molecules, the derivatization of the molecules, making starting points for everyone else in every other field easily accessible. And we have the kind of quantity, as you'll see in a minute, that will allow us to do direct assays early in this process. So when we talk about scale, we're not talking about bottle samples. Over here to the left, you see a typical laboratory sample. In fact, in many cases, this is a large laboratory sample. At 100 milliliters at a lab benchtop area, that's a pretty big sample. You can do genomics on it. You can do peak chemistry on it. You can start to understand a lot of things about it. What you cannot do is get at the cellular signaling molecules that are in parts per billion and parts per trillion because you don't have enough. So we don't start with 100 milliliter as a typical sample. We start with 200 million times that quantity. So getting at parts per trillion and smaller quantities is easy. Put that in one perspective. If we run our systems for 1 hour, we can concentrate a 350 molecular weight molecule to 50 grams in 1 hour. So we're right now at 7 orders of magnitude, not a 10 times improvement in existing technology. We're at 1,000,000 times improvement in existing technology in order to obtain the quality and quantity of biomass in a particular way to preserve all the opportunity to look at the proteins, the peptides, the small molecules, the lipids that could be products of the future. Now, where we look at the law of diminishing return, we could see this going 2 more orders of magnitude, and we envision a harvesting facility in the Chesapeake Bay and in other areas that would ultimately accumulate massive quantities of these hidden chemicals and make them available not only for industry, but other purposes.
Ross, can I jump in here?
Please.
All right, so you talk about the Chesapeake Bay. Does the makeup of the water add or detract from the number of microbiomes? So like let's say where the Mississippi River flows into the Gulf. There's a lot going on there. Would that actually increase the number, or would that have a negative impact on the number? Or maybe closer to a port where there are a harbor where there are a lot of boats and ships and a lot of things happening. What does that do? Does that increase or decrease?
So when you talk about super clean water, if we were to go to the mountains of Colorado, you've got very little nutrients. Therefore, you've got very little growth of microbes. In and around cities, Chesapeake Bay, the Indian River, the Gulf of Mexico, there's areas where you're going to be producing what is a lot of nutrients. It's called eutrophic waters. And in these eutrophic waters, you have a dynamic of warring molecules. Now, the interesting aspect of, let's take a Chesapeake Bay, is there's many different inflows, so various nutrient contents. There's various salt concentrations. There's various temperatures. There's various oxygen content. There's different pHs. There's different phototrophic zones. So the reality is you have massive areas where microbiomes that are very different exist. And every microbiome, just like a human microbiome, what we've seen is virtually every microbiome in the aquatic environment has major differences that could be interesting to explore. But this is the kind of thing that has never been done.
Thank you.
One of the things that I'll point out here is you can explore things genomically, but the problem is you don't know really what's being made by the genomics. You go back to that Petri dish, that large Petri dish example video, and you see that the same species of E. coli is there in all of those areas of that Petri dish. And they would all measure as E. coli, and they would have virtually overlapping genomics that would say they're E. coli. Yet you could see from that alone, they do very different things. That's one of the challenges with microbes, is microbes of a given species might actually be 30% or more different and still be called the same species. And when you look at something like that, as far as that diversity, if we're measuring just the microbes in genomic senses, and we're not fingerprinting their molecular output, you really don't know what you're looking at. And that's one of the challenges that comes with potentially monitoring microbes. You can certainly use genomics to monitor it, but it's not going to tell you what's really going on. And that's one of the challenges. So when you talk about what we do scout and analyze a site to make sure it's got the genomics. We use genomics. We're supporters of genomics, and we're supporters of closing the metabolic pathways to the actual molecules, because that will really advance science. So we use genomics and we use chemistry to do analysis. We select a site, we get approval. What's really important here is we pull out the microbes and we put back clean water. So we've been approved to do this even in a marine sanctuary in California, where we did a scout harvest and got state, local, and federal approval in that marine sanctuary. This is environmentally friendly harvesting. And even if you look at the massive scale we're talking about in the future, it will have virtually no impact on a habitat, because microbes can repopulate it about as fast as we can take it out. We typically harvest right now 20 million liters, about 8 Olympic-sized swimming pools. And when we pull it out of a living, breathing microbiome, we do it without lysing or breaking any cell. And then we preserve it. Basically, it comes completely through our system, is dewatered in the process, and 2 minutes and 14 seconds later, we start to preserve it. So we are not looking at sludge. We are looking at a living, breathing microbiome, and we're locking in the chemistry of that habitat right then. Our next step in our process is lyophilization. And when we lyophilize, we then go through a series of extractions, a series of fractionations, and then the analysis starts. And this is where we use a number of machine learning and AI tools. For instance, this weekend, one of our AI tools was able to look at one of our microbiomes and found what we think is a significant finding, and that is science knows that a compound structure skeleton called indole is very bioactive and creates all sorts of opportunities for therapeutics and other products, including agrochemical products. Out of indoles that are known to come from the natural world, only 4,100 are listed or exist. In one microbiome, we believe we found another 1,800 novel indoles. These can become starting points for what is bioactive molecules to go into the life science pipelines or starting points of the future. So we use AI to help understand what we've got before we ever do structural elucidation. We then prioritize using ML AI so we can curate it and bring it to our partners. Now, our partners, as I showed in an earlier slide, would then screen that, find hits against targets. We would work with them to build the IP portfolio, the derivatization around it, and then they would advance it through their channels, whether this is a pharma company, a biotech, whether this is an agrochemical company, or whoever needs to find these kind of products. When you talk about the depth of discovery, right now, this little red dot, which might be very hard to see on some people's screen, I'm trying to put my cursor on it, it's about in the middle of the screen, is today's depth of technology of an uncultivable microbe with an unknown small molecule. So to find an unknown small molecule and an unknown microbe is very difficult, and it takes a lot of effort. In our particular case, we scale up dramatically, and we're already at 7 orders of magnitude beyond the typical capability. Now, that was a lot. I'm really interested in answering questions about why microbes, why the data of microbes, why the molecules, why our technologies, before I jump into what are some of the possibilities. Because I've got a number of possibilities around what would be material opporttunities, what would be energy opportunities, and what would be defense opportunities. And before I dig into that, I was hoping to answer just a little bit of questions about the core technology or the reason why this is so game-changing in the world of life sciences and how it'll positively impact humanity. So I don't know if there's any questions.
Hi, Ross. This is Abigail DeWalt. I'm from Ditcha. I'm really interested in hearing a little bit more about your use of AI and ML and lyophilization. Unfortunately, I have to drop off this call right now, but would you be able to drop your email in the chat just so I can follow up with you a little bit further on this?
Yes, and I'm on LinkedIn, and I'm Ross Youngs and Microbiome. You'll find me really easy.
Okay, great. I will be reaching out to you soon. I'm really excited to learn more.
Terrific. Thank you so much.
Yeah, Abigail, I'll respond to your email you sent earlier.
Thanks, Eugene. Everyone, this was really great. It's sad to leave, but thank you for a great presentation.
I appreciate that. Thank you. Any questions? This is the challenge when you have a brand new technology that nobody's ever really utilized. People may be thinking, well, how in the world would I collaborate with Biosortia? What kind of products could we potentially find? That's really why we're excited that Eugene invited us to this forum, and we're looking forward to connecting with people to help them solve those kind of problems in the future. So any questions about the technology or the reasons why we are where we're at?
Ross, hi, my name is Mackenzie McAleer. I sent you a question there in the chat about how your technology can be scaled for creating beneficial biology for agriculture.
Yes. Yeah, I think one of the big areas there is there's been really no true new product classes in agrochemical for, I think it's 24 to 28 years. It's been a very dry area. 98% of the patents are off patent protection. And the problem we keep running into is life forms will evolve to not necessarily react to the chemistries that are in place. And then we're learning things about chemistry that are in place that are forcing us to remove chemistry. So there is a massive opportunity to find next generation agrochemical products. And if we look at the history over the last 28 years, we've had AI. I mean, people are acting like AI is absolutely brand new. AI was touted, available, and used, and a lot of analytical capability has been used for decades. So we've had the tools, we've had genomics, we've had synthetic chemistry, and we are not finding new products. The best place to look for these new products is what nature has actually done to create the molecules that hit the targets. So we think going to the microbial world will open up a massive opportunity for next generation products. One of the biggest objectives out there is how to find a replacement for glyphosate. Bayer announced that they were going to put $1 billion toward finding that replacement. Are they looking at industrial scale microbiome mining for pulling the hidden secrets of dark matter of biology and microbiology out? No, they don't know about it. That's why we're starting to talk about is to make sure the scientists of the world can engage us and say, what can we do to advance this technology? Is there anything specific I can answer? I tried to go general on purpose, but is there something you're thinking that needs to be solved that this kind of technology should be able to help?
Essentially, the ability to curate the microbiology in soil or to produce beneficial biologicals for consistent soil production, particularly indoor production, is what I'm interested in. But when you think about prescription produce, there's a big right now in the medical benefits and the medicinal effects that a lot of really standard crops can have. And there's some work being done on the microbiology in soil and that in your gut, and essentially finding the communities that are the most beneficial and the best ways to grow crops that have wellness enhancing effects.
Yeah. No, those are really good points and really good questions. I'm personally not a real big, I'm not saying it's not possible. I'm not saying there's not going to come useful products from it, but I am not a real big supporter of, quote, bugs to drugs or bugs to products. And the reason is they evolve so quickly that whatever environment they're in, they're going to exchange genes with other microbes. They're going to not do what people expect them to do and will probably be invalidated or muted in their purpose rather quickly. So the other thing that happens is bugs to drugs in humans is you're talking about strain differentiation, yet it's all the same species. You're talking about potential viral contamination or phage contamination. I think it's going to ultimately be useful in the future, but we better be able to fingerprint what is really a habitat and an opportunity and look at the molecules, whether we're fingerprinting the humans itself and its ability to produce the metabolites that enter the bloodstream or the serum and get through the blood-brain barrier. We need to be able to understand exactly what's going on. And right now, we don't have a clue. So throwing a microbe at something might solve some problems, but I would think we will be better off when we can have a clear understanding of what's going on within a particular target area. And that's why I would say that getting to the actual molecules, understanding the proteins, the peptides, the lipids, digging deep into the dark matter of biology and the microbiology is going to be part of the process of solving all of these problems and probably a critical part. Any other questions before we dig into randomly picking on some of what we have here?
Roy, you've been unmuting.
Yeah. Can you hear me?
We hear you perfectly.
Yeah. Okay. I have a question. What's the output? Is it a protein sequence of these different chemicals, or?
Yeah. So when you have access at this kind of scale and you process it properly, you actually get the molecules, whether these are going to be the enzymes that proteins produce. And there's supposedly at least 1 million enzymes that are unknown that are produced by microbes, which will probably play heavily in future products, future technologies. When you look at the small molecules that are produced by microbes, in just one analysis of a microbiome we've played with, 73,000 unknown small molecules were in just one natural microbiome that we have access to. So it's data that fills in data gaps, and it's data to be used to accelerate discovery and prioritization. And then it's the actual access to the materials that can be directly tested. That's unlike the indirect technologies that are dominant across all of life sciences today. The indirect technologies do not allow you to get to the molecules nature has made, except for the direct technology of culturing, but that has been a tough way to go as well.
So are you just sorting the different proteins and saying, we've got this one that's responding to this chemistry, and we've got another one here, another one here. Then you kind of, are you segregating those molecules so that they can be tested further?
Yeah, that's actually a great way to put it. So we've kind of referred to the oil industry in a cracking tower. So just use that as an analogy. Even though raw oil comes in, asphalt comes out, gasoline comes out, diesel comes out, heptane, all sorts of polymers and chemistry come out of processing that crude oil. And ultimately, some of those products are way more valuable. In our case, we're going to the microbiomes of the world that have been virtually untapped by life sciences, creating a super cracking tower to get at these materials and present these materials to research, understand, analyze, create the data sets around them, do the predictions, do the target analysis. So it is about getting at the material, which is primary, but it fills massive data sets. I hope that helped a little.
A little bit, yeah, thanks.
And when we put it in perspective that we're going to harvest 20 million liters at the scale we're currently at, and out of that will be 1,000,000 grams dry weight of that microbial community. That microbial community represents a significant amount of proteins. The next largest component's lipids, and the next largest component is actually the small molecule. So we're in a position where having that material in hand and in quantity means if we get an activity, we have enough of it to go find where that activity came from, especially if we're doing screening. And we've been able to prove that. And I think Polyplexus was going to post some of our papers in references to what we're doing. But at the end of this, we're going to put our contact information up, and we really encourage people to reach out to us because we need to find early projects. We need to find early partners. We need to find people that want to fund this advancement, whether it's through investment or through grants or through potentially donations. We need to move this technology forward. We believe what we've learned suggests this is going to be critical to humanity in the future, because if we look at crop utilization and needs to improve productivity, if we look at health of the human and other species, if we look at our own environment, how are we going to stop red tide? The best way to stop red tide is understand it and fingerprint it molecularly, look at it before, during, and after its bloom, and find out exactly what could inhibit the brevetoxin from being produced or the species from dominating the environment. When you look at coral bleaching, understanding molecular aspect of what's going on could create solutions. This is a critical technology that nobody knows about, and it's going to take engagement to move this thing forward. It could be 10 years from now, 100 different universities and research centers are all doing industrial scale microbiome mining. It's the right way to get at the hidden molecules of life. Any other questions?
There was another gentleman that was on, but I don't see him anymore. Sanjay, he had unmuted a couple times.
Terrific. Well, I don't mind being interrupted.
All right. So my question, Ross, how do we make this happen? Let's get into that.
Well, I've got a book on my shelf that talks about how hard it is, and I'll hold it up in front of the camera. It's The Conduct of the Inquiry, written by Abe Kaplan in like 1964. And he, about the same time as Maslow, came up with, if all you have is a hammer, then everything is a nail. They kind of simultaneously did that. Kaplan was writing about the life science industry back in 1964. And one of his biggest issues was if you have to get a PhD, or let's say you don't have to, you really want to get a PhD in something, you study it and you learn more and more about that subject. You become an expert in that. You promote that. You write papers on that. You write proposals and funding things on that. You get hired by people to do that. When you're talking about a breakthrough novel technology like this, none of those people exist. So it takes political power and will, and it takes the understanding of forward-leaning organizations to say, this is so important, this is so critical, we need to move this technology forward. But the problem is a lot of the people inside that were trained in other subject areas and promote other subjects. So it's a chicken and egg dynamic, and nobody's going to do it until somebody does it. Kind of like the electric car and Elon Musk. Would we have an electric car today if it wasn't for Elon Musk saying we're going to do it? And look at what he had to invest in order to get it there. We'd like to think that this investment is much smaller because, quite frankly, we've got ready-made customers across the entire life sciences world that have been struggling to find starting points that are real and pre-optimized by nature. There are many pharma companies out there that will say, we got all the molecules in the world we need. No, you've got all the wrong molecules in the world you need. There's one Chinese firm that I met at JP Morgan that has produced 2.5 trillion synthetic biology molecules. The reality of it is none of those represent what nature actually does. And getting to nature is uncovering the secrets so we can use it. There are many examples of biomimicry, bioutilization of products across the entire spectrum of life sciences. This is just a better, faster, and actually cheaper way to do it that no one has ever been able to do, because there were barriers that have been eliminated. Anything else? Should I pick one of the ones that are on the screen here and talk about it a second? When you look at trying to ideate possibilities, I think one of the things Eugene said to me is fish don't end up fouling and microbes stay off of fish, and they can swim around in the ocean for an entire lifespan, and yet we can't create a ship or a dock or anything and put it in the water and not have things grow on it. So studying the properties of, and it's down here at number 19, studying the nonstick properties of certain microbial biofilms could inspire the development of surfaces resistance to biofouling or marine growth. So whether we study a fish or we study the microbes or we study the enzymes in the metabolites and turn those into products, the idea that that could happen is real.
Yeah, thank you. Yeah, that is the goal here. The goal here is that, what I found when I ran this federal version of this for the federal government for 7 months, we ended up setting up over 400 collaborations with just 52 presentations. And the beauty of it was that so many of these applications were outside of where the manufacturer, where the developer of the technology saw application. And it was by bringing in people from the different areas